Okay, so this video is a short version of DNA replication, focusing mainly on the enzymes required for replication. So let's go ahead, and here's a list of the enzymes I'll be talking about. So topoisomerase, helicase, primase, DNA polymerase 3, DNA polymerase 1, and ligase. I recommend, if you are a student studying this, to maybe pause the video after I talk about each of these and take a little bit of notes on what they do. Okay, so let's go ahead and start with the double helix here. So we have two strands of DNA. Now it is a tightly coiled helix, so we are going to need an enzyme called topoisomerase that is going to loosen that supercoiled helix, which then makes it easier for the next enzyme, helicase, to go ahead and unzip the double helix. So as a replication occurs, topoisomerase is going to loosen and make it easier for helicase to break the hydrogen bonds. So as helicase is moving down, we call this the replication fork, like where the DNA is splitting, like a fork in the road. If you're hiking on a trail or walking on a road, a fork in the road is it splits into like a Y shape. So here, the DNA, this is the replication fork where it's splitting. In this case, my replication fork is moving to the left. And so topoisomerase is right before or right around the replication fork. And then helicase is breaking those hydrogen bonds between the base pairs. There's 2 hydrogen bonds between an A and a T, and 3 hydrogen bonds between a C and a G. So anyway, so this is the replication fork is moving to the left, and let's go ahead and see now what some other enzymes are. So the goal is to replicate DNA, so we have 2 identical copies. So the cool thing about DNA replication is that we have base pairing rules. If we know one side, we can build the other. So to do that, we would normally use DNA polymerase 3 is able to build DNA in the 5' to 3' direction. So unfortunately, DNA polymerase 3 is not able to stabilize nucleotides well enough to begin to just start attaching DNA nucleotides. So we need to put a primer to kind of help the process. So we have a different enzyme called primase. Now primase is going to build the RNA primer. So here I have RNA nucleotides that are going to be base paired with DNA. Now primase is able to stabilize the nucleotides well enough in order to attach them together to form these primers. Now you notice they are being built in that 5' to 3' direction, and because the replication fork is moving one direction, the DNA is actually built in opposite directions due to its anti-parallel nature. But I have a whole other video on replication. This is just supposed to be on enzymes. All right. So here, primase is going to build the RNA primers. Now, once there are primers, then DNA polymerase 3 can come and continue elongation. So now DNA polymerase 3 can attach a DNA nucleotide. Now you notice the A is base paired with T, and we're adding on to that 3' end of the primer right there. So DNA polymerase 3, when it is building in the 5' to 3' direction, if it is on, this is what we call the leading strand. If it's moving in the same direction as the helicase and the replication fork, it will just continue and it'll build this strand continuously. So it'll just continue and follow along helicase and topoisomerase building continuously. Now the top strand, though, is built in the opposite direction of the replication fork. So therefore, DNA polymerase 3 will attach on to the 3' end of the primer and do base pairing rules and then build the fragment in the other direction. So we have here the leading and the lagging strands. So the leading strand, like I mentioned before, should be built just continuously. However, the lagging strand, we've opened up. So in this section here, we want to think about how the replication fork has continued to unzip. So now there's a new exposed area. So now a new primer will be built with primase out of RNA nucleotides. Now DNA polymerase can come and fill in that new section of exposed DNA. So we have another fragment of DNA here on the lagging strand. So when we're done with this, we have two identical copies. We have our original strands and then our new complementary sequences. However, there's these fragments in the lagging strand that we call Okazaki fragments. We cannot leave these RNA nucleotides from the primer within these fragments. And then also on that leading strand has some RNA nucleotides we also need to remove. Now comes DNA polymerase 3. DNA polymerase 1. So DNA polymerase 1 is going to come and remove these RNA nucleotides, and then we'll replace them with DNA nucleotides. So it also will proofread and check for errors as it scans, looking for RNA nucleotides. And then the last enzyme we'll talk about is ligase. When we replace those nucleotides, we need to build the phosphodiester bonds. So ligase will come and build a covalent bond in that sugar phosphate backbone. That covalent bond is called a phosphodiester bond. So ligase, you can kind of think of it like tape or a welder. It's going to come and like weld the sugar phosphates together of those fragments. Now you notice down here in the leading strand we have some RNA. So again, DNA polymerase 1 will remove that, but unfortunately it cannot add on to the 5' end. So every time our DNA replicates, it gets a little bit shorter. And that's why we have telomeres. Okay, so now let's go ahead and double check that you can explain each of these 6 enzymes used in DNA replication. All right, Rick, great job.